Very good evening to all. I'm Dr. Pankaj from JSS College of Pharmacy, UP, also convener for Scientific Committee, Pharmanas 21, which is organized by Pharmacy Federation. So today is the last day of poster presentations. So since last 1 week, we are completed many presentations. So I welcome today's evaluators for this presentation, Dr. Subhas, Dean School of Pharmacy, GH Raisoni University, Madhya Pradesh. Welcome you, sir. Dr.
Thank you,
Madhav
sir.
Divedi
Yes,
from
sir.
Gitanjali University, Udaipur. Welcome you, sir.
Thank you, sir.
Dr. Shankar, Professor, PhD College of Pharmacy, Coimbatore, Tamil Nadu. So we welcome you all and let us start the session. Sweta,
Thank you, sir. Now I would request eNest 449 to come up with your presentation, and I would request Chetan Mehta to show the eNest poster 449.
Aman, it's visible?
Yes, it's visible. eNest 449, you are not audible. Please unmute yourself.
Yes, sir. Am I audible?
Yes, now you are audible.
Thank you, sir. Pleasant welcome to one and all present here, judges, OPS members, and Vikram Chaudhary, sir, and scientific conveyors, to give a wonderful opportunity to present my e-post on this platform. And my name is R. Shiva Armugam. I'm studying in JSS College of Pharmacy in B.Pharm final year, and my poster topic is designing and my poster topic is designing next generation epitope vaccine against the pathogenic strain of Pseudomonas aeruginosa. And my aim and objective of this is to produce an epitope vaccine against Pseudomonas aeruginosa and evaluate its safety and efficacy. Introduction. Pseudomonas aeruginosa is a confined rod-shaped gram-negative bacteria which acts pathogen for both human and animals, and the main core genome is nearly 6.3 MB, which facilitates in multi-drug resistance, and the mortality rate seems to be around 18 to 61 percentage, and the multi-drug resistance is nearly up to 22 percentage. There is no vaccine has been approved against Pseudomonas aeruginosa, and it mainly forms a biofilm. Biofilm by quorum sensing and EPS, extra polymeric substances and external genomic substances, so that when it combines to other microorganisms, the extra genome substances are obtained by the other microorganisms, causing various drug resistance. So this gave me an idea to create a vaccine against this. Even though there are some substances that can kill Pseudomonas aeruginosa, I targeted to create a vaccine is to provide against the multi-drug resistance caused by the Pseudomonas aeruginosa. The Pseudomonas aeruginosa can cause several infections in open wound, fire wound. It has been constantly reported by the WHO over 60%, and these also these forms biofilms in the catheters, ureter catheter, like that instruments, and causing mortality. And I have used in silico studies, and in methods I have obtained the sequence from the NCB dataset, and I have evaluated its stability. Then I have gone to in silico testing. That in silico testing includes the stability testing, antigenic testing, homologous testing, and Ramchandran plot test, and NMR and X-ray scopic test. And then I have constructed the vaccine, and then vaccine is subjected to its stability again. So the vaccine should be stable, and then I have gone for the immunostimulation, and the immunostimulation report has shown a good report. And when we give this vaccine, it can inhibit Pseudomonas aeruginosa in our body. Results. The immunoglobulins has been raised, and I have given nearly 1,000 units. The accurate unit is 989 units for 60 days, and I have produced the result. And in NMR and X-ray spectroscopy, it found to be a good vaccine because it goes inside. And I have gone for the primary and secondary structure in SOAPMA server. The primary structure is so good, and the secondary structure also supports with it. And then I have checked the molecular dynamics, the deformability and B factor and the eigenvalue and integration are good. The integration is more correlated amino acids than the uncorrelated amino acids. And finally, I need to discuss about the vaccine. The vaccine has the following characteristics like antigenic property, because every epitope I have selected is an antigenic and non-allergen and non-toxic. Because of I have used some linkers with adjuvant like GLP, and that I have found that G score as minus 7.38, and it shows high stable. And then I have done the immunostimulation, which shows some greater efficacy. Therefore, I conclude that the constructed vaccine is a good vaccine candidate for the Pseudomonas aeruginosa. And I wanted to thank my beloved professors and OPF members and the judges and scientific conveyors and organizers who gave a wonderful opportunity to express my views. And I thank you, judges, for patiently listening my e-post topic. And thank you, sir.
Thank you, Ines 449. Now I would request the evaluators to kindly come up with the questions.
Yeah, this is Dr. Shankar. What is the need for specifically for Pseudomonas aeruginosa you want to generate?
Good evening, sir. Sir, my focus was to develop a vaccine against Pseudomonas aeruginosa against all other is it creates multi-drug resistance when it enters into our body. Our body contains.
What are the diseases specifically it is responsible? Can you state that one?
Sir, it mainly forms biofilms, sir. The biofilms, that biofilms include several microorganism colonies, which forms and it creates toxicity, sir, cytotoxicity.
Okay. The biofilm is there and a lot of antibiotics are showing resistance. So how you were strained, how you were simulated one will overcome the biofilm, what steps you have taken to overcome the biofilm problem?
Sir, biofilm problem. Okay,
Yeah.
Sir, whenever our antigen enters into our body, our CD8 plus cells and CD4 plus cells will go and our TLR receptors will analyze and will give signals to CD8 plus and CD4 plus cells and B cells. And whenever there is when an antigen enters, it analyzes and produces, it stimulates the NK cells, or it produces antibodies by the B cells. When an Pseudomonas aeruginosa enters into our body, before forming the biofilm, it can kill. Even if I have an hypothesis, sir, based, I have to check, but I have an hypothesis. Shall I say, sir?
Yeah, okay, no, that is fine. So these epitopes, what epitopes you have taken for your study?
Sir, I have predicted CD8 plus epitopes and CD8 plus epitopes, CD4 plus epitopes, and linear B cell epitopes, sir.
Yeah, so you have to take the biofilm protein epitopes also, because your aim is to make mainly the biofilm, that type of protein epitopes also you have to take it. And along with then without that epitopes, how it is, what is that, in vitro, how it is behaving, that you have to strengthen that. Okay. Because nowadays not only the antibiotics alone cannot overcome the antimicrobial resistance. So antibiotics designed with the antibiofilm agents is the need for the hour. Okay, so you take the protein epitopes of biofilms also. Okay,
Sir, it has been included, sir.
that also
I
included.
have got it from, yes, sir. I have got that anti-biofilm peptides from DHPB, sir. So,
Yeah, that's my question. Yeah, that's what. Okay, thank you.
yes, sir. Thank you, sir.
So have you tested those protein epitopes?
Yes, sir. I have tested that protein epitope for the above categories, like antigenic, non-allergenic, non-toxic,
So-
stability, and homologous, sir.
So what was the result?
The result was every peptide seemed to be antigenic, sir. It is mostly predicted, so that the average score will be around 95 only. We have to test it in vitro. It also doesn't increase these levels of mast cells. So I conclude that it is a non-allergic. So we have tested this toxic from Swiss Prot references. So it shows less toxic only. So when we do in vitro only, we will confirmly know that how much toxic it creates. But according to in silico, it's lesser, and it's highly stable, which is evaluated from physicochemical properties. Sir.
Okay. Thank you.
Thank you, sir.
Okay, could you tell me any clinical trial is going on to develop vaccine for this gram-negative Pseudomonas aeruginosa? Clinical trial.
Yes, sir. Sure, sir. Sir, there are some vaccines under development, sir, but there are nearly 4 to 10 vaccines which are under development, but no vaccines yet has been approved by FDA, sir.
Okay, could you tell me one question? Which antibiotic is very active to treat this Pseudomonas aeruginosa currently? Which antibiotic? Means you told that there is a multi-drug resistant because this particular this particular bacteria is getting resistant with multiple antibiotics. Which one is currently you can say that is antibiotic which can 100% efficacy on this Pseudomonas aeruginosa, because this is very severe infection for the lungs.
Yes, sir. Yes, sir.
Tell me.
Sir,
Which
there
antibiotic?
isn't any drug which can give 100% efficacy against Pseudomonas aeruginosa, sir. There are several strains which have been identified and yet to be identified. But now they have been for biofilm to wash that urethra catheter-like things. They are using ethanols and some penicillin-based drugs they are using, sir.
Okay, could you hear about cholesterol? Cholesterol. Cholesterol.
Yes, sir. Yes,
Cholesterol
sir. Yes, sir. Cholesterol is also being used.
is also there. Okay, thank you, dear.
Thank you, sir.
Okay. Thank you, sir. Thank you, Ines 449.